A subject very dear to my heart. I've just come back from France yesterday from the International Campylobacter Conference, and as you can imagine, there's a huge interest in what's been happening in here, in particular the Havelock North incident. So what I'm going to do is just give an outline of the current situation in terms of a few waterborne pathogens, what we know about their epidemiology in people, in animals, and what we found in water. Then I'm going to talk about the application of molecular typing, so how we can make the link between animals, water, and people, and then a focus of what we did, particularly around the Havelock North incident. So there are a whole number of different waterborne pathogens. The ones I'm going to focus on are Cryptosporidium, Giardia, Campylobacter, and we've got E. coli 0157 down the bottom there, but there are a number of other pathogens that internationally are recognized as being important. Now, many of these pathogens are also foodborne pathogens, so I have an interest in food safety. There's a very strong parallel between food safety and water quality and water safety. And this is from the ESR League table of notified diseases, and you can see right at the top there, Campylobacter, much of it foodborne, and a few other pathogens there which have a food and waterborne component to them as well. So just talking about Cryptosporidium and Giardia, what do we know about those? Well, one thing we do know is that our notification rates are relatively high compared to other countries around the world. This probably reflects our pastoral livestock system, our high numbers of carrier animals compared to human beings. And this was a report reported in Epidemiology Infection paper, which showed our rates relative to other similar OECD countries. It's highly seasonal. Cryptosporidium is highly seasonal. You can see outbreaks here. Every springtime we see an increased incidence associated with one particular species of Cryptosporidium, Cryptosporidium parvum, and then every now and then we see increased outbreaks in the spring as well. You can see one here, associated with Cryptosporidium hominis. Now, hominis is the swimming pool, human-to-human one. Parvum is the animal, particularly livestock reservoir to people. And Giardia, you don't see so much of a seasonal pattern to Giardia patterns over time. So the two species that we're dealing with Cryptosporidium, the ones that come from cattle and sheep, and this is from our survey data, and the blue bars here, Cryptosporidium hominis, which is the swimming pool and person-to-person associated one. When you look at the spatial relative risk maps, you can see a high relative risk, particularly in rural areas for Cryptosporidium. You can see in strong association with dairy farming areas, Waikato, Taranaki, South Canterbury, whereas not so much of a rural association with Giardia. So very different patterns you see between the two pathogens. A couple of papers that have appeared recently have done a lot of typing, looking at Cryptosporidium and Giardia genotypes and what we know in New Zealand. And just to summarize this, so Cryptosporidium hominis, which is the person-to-person one here in New Zealand, we have type 1b, and in Cryptosporidium parvum, this is the one that's really strongly associated with ruminants and with people. And we have one particular strain here, which is the most common subtype found in people, and also the most common subtype found in cattle and sheep. And this is quite unusual. This is a genotype that's only found really predominantly in other countries like Ireland, but relatively rare throughout the rest of the world. With Giardia, although there's a common belief that Giardia is a person-to-person spread pathogen, we also see two of the strains of Giardia, which are most common in people, are also found in cattle and in ruminant sources. Whereas we have this one here, which is type E, which actually is very common in cattle and sheep, but doesn't turn up in human cases. So what about the prevalence? So how common are these pathogens in feces of cattle in particular? So I'm focusing on cattle, a couple of publications that appeared in the last couple of years. This was a survey that was done in 2011, over 1,000 samples from 97 dairy farms across the whole of New Zealand, or the main dairy farming areas in New Zealand. And essentially what we found was the prevalence on farm, particularly in calves, so these are young calves under 5 days of age, and these are calves up to 3 weeks of age. Over 50% of the farms were positive for Cryptosporidium parvum for these calves here, and slightly less for the younger calves. Giardia, almost 90%, but most of the strains of Giardia were those ones that don't cause disease in people. At the calf level, again a difference, so very young calves, about 6% were positive, but for the older age group, about 16% were positive. So this is a prevalent pathogen on dairy farms throughout New Zealand. So just to put, this is a slide that I borrowed from Brent Gilpin from ESR. I'd like to acknowledge a lot of the work we've done in collaboration with him. The number of defecations a day, this gives the scale of how much fecal material is being produced by our cattle population. They do it between 9 and 16 times a day, so a bit more frequently than we do. A bit of greater volume as well, 2 kilograms. Total output of around about 25 kgs per cow per day. Now, if you multiply that up by your 9 million cows, that's 230,000 tons of fecal material which is going into the environment every day in New Zealand. That's 84 million tons a year. Now, as was mentioned earlier, humans also in New Zealand produce 800 tons a day, but that's treated and so it doesn't represent the same threat to the environment. So just moving on to what we find in water now. So abstraction points, we've been sampling a number of 20 abstraction points or source waters from different sites for the Ministry of Health over a number of years now, about 540 samples that we've looked at. And of these sites, the groundwater sites, I've covered up the labels here, but the groundwater sites are these ones here. So we don't find Cryptosporidium or Giardia in any of those groundwater sites, but we find up to 80% of these sites up here, in particular Waikato sites, Waikato River sites, positive for Cryptosporidium and Giardia. So it's certainly prevalent in those source waters. And some of the concentrations would need a log 4 or even log 5 credit to reduce them down to a safe level. This is an earlier study. It was reported by Des Till, reported about 8% Giardia and 5% Cryptosporidium. So these are kinds of numbers that you can get in your source waters. So moving on to a different pathogen now. So talking about E. coli 0157 or Shiga toxin producing E. coli in general. Now this is the one that really, really worries me, and I know it worries a lot of people, and it's been mentioned a couple of times as well. So I'm going to ask the question, how prevalent is STEC in general and 0157 across New Zealand, and why should we be worried about it? Well, we know that E. coli 0157 and other STEC are associated with cattle. They can cause severe bloody diarrhea, a high prevalence particularly in preschool children, and particularly in children living in rural areas. But they also can cause death in a number of cases, and about 8% develop severe renal failure, severe kidney failure, and other complications associated with the infection. And what we see in New Zealand, the highest prevalence of human infection is strongly associated with our dairy farming areas, so Waikato, Taranaki, and South Canterbury again. We are seeing a gradual increase in the number of cases. Now, this quite dramatic increase is probably an artifact as a result of changes in testing to move towards non-culture-based testing. But nevertheless, over a 10-year period from 2004 to 2014, we saw an increase in the rate of over twice over that time period. And the more recently, because of the use of different testing, we're seeing a relatively fewer of them as 0157, but still nevertheless 0157, which is probably the worst one, is still quite prevalent in human cases. And our rates are relatively high compared to the rest of the world. A case control study didn't identify any food-related risk factors. This was published in 2013. But what we did identify was contact with farm animals, contact with cattle feces, and contact with recreational water, and also drinking water, non-main supply drinking water, were risk factors associated with 0157 and other Shiga toxin E. coli infections. So how prevalent is our STEC on dairy farms? So this was a study done similar to the Crypto and Giardia one, but done in 2014 by one of our students, Springer Brown. 102 farms, 1,500 calves, funded by government and industry, and we used a validated independent, culture-independent assay called NeoSeq. Now this simultaneously detects the presence of the virulence gene and the serotype in the same bacterium, so we can reasonably confidently use this test to determine the prevalence of STEC. And we can focus on what are called the top 7. So these are the ones that are most important from a food safety point of view, and the ones that are most important from a public health point of view. So these are the results. So you can see the prevalence of farm level prevalence varied of any top 7 between 60% and 100%, so the highest level in Northland and the lowest level in Taranaki of 60%. And the calf level prevalence, you can see here, reasonably high prevalence in those calf populations. So these are very young calves, bobby calves basically intended for export. So looking at individual ones, so these are the 2 that turn up most frequently, probably the most dangerous of the two STEC. And again, you can see STEC 0157, relatively uncommon, but nevertheless 17%, 50% of farms down here. You can see it still is quite a prevalent pathogen across New Zealand. 026, even more prevalent, particularly in Canterbury and Southland. So this is not an uncommon pathogen in our dairy population. Now, we've been doing a lot of genome sequencing work, and this has given us a really good indication of how this pathogen's evolved, how frequently it's entered New Zealand, and what's happened to the size of that population over time. And using these kinds of tools, we can say there have been at least 3 introductions of E. coli 0157 into New Zealand, probably in the latter half of the 20th century. So this is a relatively new introduction of this pathogen over that time period. The first human case was in 1993. We also saw a big increase in the number of live cattle that were imported in the 70s, and that may have been around about that time that this particular group of pathogens were introduced into New Zealand. Now, even more interestingly, what we can do is we can also model the size of the population going back over time, and we can estimate that for all of those different lineages that have come into New Zealand, they're gradually increasing population or exponentially increasing population since the mid-1980s. And this corresponds reasonably well with an increase in the dairy cattle population over that time period, and also an increase in the stocking density of the cattle population over that time. So we know that density is associated with enhanced transmission, increase in bacterial populations. It probably wasn't surprising. It took a little while before we saw the first human case, but subsequently that increase in human cases could easily be explained by the change in the population over time. Okay, lastly, I'm going to talk a bit about Campylobacter. So I've spoken about Cryptosporidium and Giardia. What do we know about Campylobacter? And I'm going to focus on the 2 most important ones, jejuni and coli. Jejuni is by far the most important of the 2. It accounts for about 90% of the notified cases that we see in New Zealand. Now, the time series looks quite interesting, because what we had was a major problem that was identified, an increase probably associated with the consumption of fresh poultry up to 2006. We identified poultry as the main source around that time, and that resulted in interventions in the industry, which resulted in a dramatic reduction. So we went from 16,000 cases a year to just about 6,000 or 7,000, and that's been maintained through to the present day. We're still trying to work with the poultry industry to reduce this even further, but you may also spot here, this is what happened to the notification rate. So this isn't outbreak-related cases, but around the time of the Havelock North incident, we did see a spike in general notified cases across New Zealand. If we look at the difference between what happens in urban areas and what happens in rural areas, this is a slightly shorter time series, but you can see the intervention in the poultry industry, you can see the strong seasonality, and then the decline after the intervention in the poultry industry. And the seasonality coincides with the barbecue season, so it's November to February time. If we look in rural areas where there's no cattle, we see a similar pattern. Whereas you look in rural areas where there's high densities of cattle, or generally a lot of dairy farming going on, you can see it didn't go away after the intervention in the poultry industry, and almost every year the spike in cases appears a few months before the spike in urban cases, and this coincides with the calving and lambing seasons. So we're seeing a different set of risk factors there. And in fact, you can model the cases by using genotyping. We can allocate them to whether the human case came from a ruminant, a poultry, or another source. And what we can see here is 2 different models, but they say the same thing, that if you live in a highly rural area, you're most likely to get your Campylobacter from a ruminant source, whereas if you live in an urban area, you're most likely to get it from a poultry source. Changed slightly over time, but that's really convincing evidence with using thousands of cases now. And we can tease out that ruminant source now because we've got better genotyping tools, and we can show for these sporadic cases that actually cattle are more implicated than sheep based on our knowledge. But you can still see the poultry coming out very strongly. This is poultry. I've just divided the ruminants into cattle and sheep sources. Obviously a very different picture when it came to the outbreak, but generally notifications, that's what it looks like. So what do we know about the prevalence in animals and water? So there's a few studies that have been done. This is one of them that I'll cite. This cattle, 40 to 50% of samples positive. Environmental water, these were recreational swimming waters, about 25%. And these are the kinds of things that we see consistently. In fact, I would argue that if you allow for imperfect test sensitivity, probably every animal everywhere has got Campylobacter. Certainly very high carriage in ruminants. And this is similar in water to what was found by Des Till. Graham McBride was also involved in that study as well. And similar to what we find in our abstraction points, we find slightly higher levels, but none in groundwater. Why is it important to public health? We can see a lot of association with chicken, but we also see these particular strains. Each of these is a different strain type of Campylobacter, and this particular strain we find a lot in water, but very rarely in human cases. Now, these are ruminant sources. The ones that we find here, these unusual water ones, actually find out ultimately that most of these are associated with Pukeka, or the water rails. So these are Campylobacter jejunis, and this is, if you do an attribution to water, this is why you find that most of the Campylobacter seem to come from water birds rather than from cattle and sheep. But nevertheless, the important thing is that Pukeka strains have not been found in human cases, to my knowledge, whereas cattle and sheep ones have. Okay, drinking water outbreaks. We've had a lot of outbreaks over time. I'm just going to focus on the Havelock North one in a second, but just to mention before, there are ones that are relatively hidden. This is one that we found through our Sentinel site, looking at the Paiatua region, and this was a heavy rainfall event, a cluster of cases, a smaller number of cases, just 9 cases. The EpiServe was reporting that the drinking water was muddier than usual, smelt of dirt. The water had been ground, so there was clearly a link with the epidemiology there. And in fact, if you do the source attribution, you found a very, very strong association with ruminants. Okay, so the final minutes or so, what do we do in the Havelock North outbreak? So this is working very closely with ESR. 2 methods were used, 2 different typing methods, one a cheap and quick one, which is the low resolution M-bit typing, and then high resolution whole genome sequencing using isolates from the different sources. Now, huge numbers of samples were taken and subjected to that first typing method, and in a nutshell, what was found was there was a strong correlation between the main genotypes that were found in people, as well as in the sheep, and the reticulated water and the bore water. So these big strains here in particular that were found that had that common association. This was using a low resolution M-bit typing, but nevertheless it was a very useful first screen. Following that up with whole genome sequencing, which is the highest resolution you can get, 120 isolates, basically 80 patient samples, some from cattle and sheep in the region, and also from the different water sources. Now again, in a nutshell, just very nearly finished, 1 minute, we found using low resolution typing based on the genome sequencing attribution to what's called a 7-gene scheme, found that the human cases again fell into these same clusters as the isolates that we found, particularly from sheep and from the water supply. The water and sheep ones grouped together when you compared them with historical isolates belonging to the same group. So very, very tight clustering of those isolates from the sheep and from the water. And when you added in the human isolates, there was virtually no genetic distance, or even no, in some cases, not even a single nucleotide that was different in the DNA sequence between the human cases, the reticulated water supply, the boar, and the sheep. So this is about convincing from an epidemiological point of view that there was a direct link between those different sources. And again, very different from these historical isolates here. So another strain, very similar finding, little or no genetic distance between them. So summary of that, basically very strong evidence, patient isolates mixed with water, ovine isolates, strongly suggesting that likely origin or confirming the epidemiological evidence, distantly related to historical isolates. So most likely multiple lineages caused by multiple clones. So last slide, summary. Waterborne pathogens, as you can see, highly prevalent in livestock in New Zealand. Surface waters frequently contaminated, particularly with ruminant-associated pathogens. Sugar toxigenic E. coli are the biggest concern due to their severe health impacts. And if the Havelock North supply had been contaminated with Aztec 0157, as has been mentioned before, it would have been far more serious. Increasing stock numbers and density is likely to increase transmission, and combine that with adverse weather events, likely to exacerbate that transmission. New tools that I've mentioned here, very recently been applied, so just in the last couple of years we can apply these new tools, and they're enabling us to make much more robust inference on the likely source of human infection, particularly in these kinds of instances, as we saw in Havelock North.